cancer, we're talking about abnormal cells that then begin to divide without control, and then they're able also to go into other tissues. And this is actually really the hallmark, the most important part of the definition of what cancer is, with this first piece, abnormal cells that divide without control. That is the biggest piece of information that makes a cell cancer versus just a little abnormal. It's also then able to invade other tissues, and it has a great disregard for the rights of surrounding tissues. And so it's sort of like the worst neighbor that you could ever hope to live next to. The name comes from the Greek word carcinoma, if I'm saying that right. So if I have any Greek scholars out there, you can give us the true pronunciation of that. But it really means crab, and then oma meaning tumor. Now, as we're going through the cancer information, it's closely tied, many aspects of it, to the changes that we see in inflammation and in the wound healing process. So when we see tumor here, it has a similar meaning. It's really actually derived from that meaning that we had in our classical signs of inflammation, meaning swelling. But in this sense, the swelling is not only due to inflammation, but it's also due to new tissue growth, out of control growth, that then also begins to bulk up that area, and oftentimes, depending on where it is, can cause a swelling to occur. This is very important. So I would write these out or print this out. Being able to differentiate between benign and malignant tumors. And we'll talk a little bit more about what exactly makes them benign, what makes them malignant. But these are characteristics that would be used to determine, especially if we're doing a microscopic view of this and looking at not only microscopic, but also different tests that we might use, such as CAT scans. We might draw blood. But especially if we're taking a piece of tissue, then we would begin to see a difference between the two of these. And Dr. Newman also goes into this somewhat, but more slowly. So we're looking at the growth of them, and we just said that they have growth out of control, so they keep growing, but they grow very rapidly also. They're not closely encapsulated, and so they don't stay in a nice, tidy area, and this is part of the process that then leads them to be able to invade neighboring areas. When we look at it under the microscope, we begin to see that the original cell begins to change, and we see a lot of different types of cells in a location that they shouldn't be at, or that they're very sort of deformed. They don't look like the type of cells that we would normally see in that tissue. In cells that are benign, there is a slowing down once those cells reach maturity. Once they reach that maturity, they should begin to either, depending on the type of cell, because we know that not all cells even really continue mitosis, and think back of what those cells would be. But then also other cells, they begin to slow that process down, and they don't go through mitosis unless they have the correct triggers and the need is present. In malignant tumors, though, they continue go through that process, and even if they're injured or they're damaged cells, they may still continue to reproduce, which then just really exacerbates the whole process. And then one last one, and then I'll answer your question, Maria. So benign cells do not metastasize. So when we use the word that a patient comes in, they're diagnosed with breast cancer, and then down the road, we find out that they now have bone cancer. So we're not talking about cells that are in the same area, not next to them, not even the same type of tissue. It's now jumped into the bloodstream, and it's taken a long trip elsewhere, wherever there is a receptive area to that type of a cancerous cell, and then it will begin doing its process there. And in fact, that's one of the ways that we know where the original lesion is that perhaps it's I have a liver tumor, a liver cancer, but I'm not having really any side effects. I'm not aware that I have it, but what ends up happening is that I end up having something showing up on a lung X-ray just by chance, and a lot of times that's how we find these things. Is we're really not even looking for them. Something showed up while we were checking for something else. We were checking for pneumonia, and instead we found something that doesn't look like pneumonia. We have to find out more about it. Do the biopsy on it, and it doesn't even look. The cells are not lung cells. They actually look like cells from the liver that are distorted. And so that's the big flag to us then, that not only do I have now something happening in my lungs, a cancer in my lungs, but even worse news, it's not the type of cancer that normally begins in the lung. It's the kind that normally begins in, say, the liver. So now I've got to go check my liver, and the patient now has a worse prognosis, and we have to look at it, do a biopsy, and then find out where else would this type of cancer normally travel to. We've got to check all of those areas. And so always a poor indicator when we see that something has metastasized. Okay, so differentiation, so good question. So as cells mature, so we think about going from sort of a continuum of a stem cell that could potentially become any type of a cell, depending on what stimulates it. Once it begins down a pathway, though, and becomes a certain type of cell, then it will become more differentiated. It's not going to go backwards normally to a stem cell. It's sort of a one-way direction, except we're going to find out that cancer abuses that too. Cancer breaks all the rules. This is in your textbook, and it really gives a great example comparing the two and what they would look like. So when we say a benign, we might have a tumor. So both of these are tumors. So both of these items here, these big lumps that you're seeing, lesions, we would say are tumors. All right, and they have a swelling. There is some reproduction. It's larger than it should be, but you can see that they look very, very different. And then I've put some of the differences between the two here on this chart so that you can see this. So if somebody has a benign tumor, does that mean that it can't cause any problems because it's benign? Can you think of a situation? What could it cause as far as a problem? If it's not malignant, it can't metastasize, right? So how can, okay, good answer, Anna. So circulation issues, fibroid with pregnancies, right? Fertility issues, yes, very good. And part of the reason is, a big part of the reason is that even though they're benign, these types of tumors can actually, over time, get very large. But even if they weren't really large, imagine if you had a tumor that was in one of these little fallopian tubes, okay? So in a much smaller enclosed area. Or, as Eva is pointing out, if you have a tumor in the brain, what do you know about the brain? It's surrounded by a hard, non-expandable skull, right? And so as a tumor, and this is actually one of the big problems we have with people getting tumors in their brain, is that as it begins to expand, it's not like having a tumor in your abdomen. So your abdomen can expand quite a bit, as we all know, after Thanksgiving, right? But there's nowhere for it to go, and so it begins to put pressure then on the brain, and also you can also have things that grow, and they end up putting pressure on the blood vessels. So then that inhibits the circulation coming to whatever tissues that was going to. And so the point being, and it can press against things, it can cause pain. That's a good point. So while it's not going to escape through this basement membrane or into another compartment, it can still cause serious problems, and oftentimes we do need to have those removed because of the issues that they cause. Okay? So, would it be possible for some kind of, yes, exactly. And so it's going to depend on the tumor itself, the type of tissue it is, and if it is fed, and this is a good example because we're right here by the ovaries, right? And so if it is something that is hormonal dependent, especially estrogen, progesterone, then we might be looking at, let me go back, we might be looking at having to address where those hormones are coming from so that we don't have future problems with it. Now, you're probably going to see that more so with malignant tumors, but again, it's all going to depend on what kind of tumor are you dealing with and what is the growth factor, what is the stimulation for that. We would want to try to remove any type of a stimulant that we possibly could, particularly with malignancies, because they are very likely to metastasize at some point. So when we say benign tumors, just kind of a little further separation with this, I'm not going to ask really specific questions about this. As far as if I give you the name of something, is it a benign or is it a malignant tumor? What I would like for you to know, though, is that if we say that it's a lipoma, whether it's benign or malignant, what type of tissue did it come from? And so you can see fat cells here, lipo, okay, so adipose tissues, fat cells. And so this kind of takes you back, though, just to looking at your anatomy, and I've given you some examples here. So they're named according to their tissues, the tissues they arise from. They're going to end with the word oma for tumor, okay, so for tumor. Now, they don't necessarily progress to cancer. They do not, that's not a given when you find these, and many don't. Some are going to just go away on their own. Some have more of a potential to progress to cancer, and that's why we were focusing on cervical lesions, cervical dysplasia. We had looked at that earlier because we know that there's a greater chance that if the trigger continues, that those will go on to develop a cancer. And even finding a dysplastic cell when we're doing a pap smear or a biopsy, if that shows up on a cervical biopsy, then that's a red flag to us. This is why we do pap smears regularly. That's a big sign to us that, no, it's not yet a cancer, but they often show up near cancers because that circle of change gets broader and broader. And so we need to go back and look further and find out, is there actually a cancer somewhere that we missed that didn't show up in this biopsy? We're not going to remove the whole cervix to see if it's okay, right? That would be not very beneficial to the patient. Alrighty, so just some things to be looking at with that, and again, they can get very large, so they can be problematic. Biggest concern, though, is if we think that there's a malignant tumor. These are named a little bit different. It's not always that easy to differentiate, and so, but they're named according to the cell type. Not so much that they will add on the tissue type, but then we're going to go further into the actual cell type from which they come up. And so these are some examples of this, mesenchymal tissue being more of a stem cell type of the tissue. And so you're familiar with these. You've studied all of these in the past. So just be carcinoma in situ. You might see it abbreviated CIS, but in this, it's sort of a gray area. So it's technically a cancer. It's an early stage cancer, but it's still in that early, it hasn't started the invasive part yet. And this we see in epithelial tumors that come from glands or that come from squamous cell areas of the body. So it's specific to that particular type of location in the body. This is the time that if we're not gonna catch it in the dysplastic area time period, then we would want to catch it as a carcinoma in situ, where it hasn't gone any further. It's still very localized. And so we mentioned in our very first lecture that watching the basement membrane is an important indicator to us for different types of disease processes. And in cancer and looking at metastasis, it's crucially important. So this basement membrane holds that tissue together, it anchors it, it separates it from other tissues or from the blood supply. And what ends up happening is that soon, if this carcinoma in situ is not excised, is not surgically removed or removed in some other fashion, if that's something that's able, usually we want to cut these out. We want to remove them and we want to send them to the lab and have that tissue examined. But if it has not yet broken through to the basement membrane, then it is not yet considered a true malignancy. So again, it's sort of a gray area here. Okay, so, and it also then has not gone into what we would call the stroma. So the stroma is the area around the cell that begins to start changing. We'll look at a picture that you have from your textbook, and this area is it's part of this tumor, but it's not strictly cancer cells. And so they all have the potential of being invaded by the cancer cells. We'll look more into that, but it's pretty stable, it's localized, it hasn't really taken on a life of its own yet. Several things can happen with a carcinoma in situ, and don't apply this to true malignancies, because once we get to that stage, it really does not go backwards. So they could stay that way, and maybe we'll say watch and see. We tend not to do that because we never know at what day or hour this is going to flip over and begin to become invasive. So I think you're going to see more. The practice is that we go ahead and we get these out. Why take the chance? Why wait 6 months and wish we had done the surgery? Okay, so just not worth it. It could go on to become invasive, metastatic. It could get smaller and just disappear. So it really kind of depends. And Dr. Newman's going to show you a lesion to watch for that's actually on the skin. And when I look at that little lesion, I just think, well, that just doesn't look so bad. And it turns out that maybe those are of greater concern than I realized, potentially. So when we're looking at cancer, you're not going to walk away. This is not even the tip of the iceberg. We're just going to find out some general information about how cancer works in the body. So it's very vast, it's complicated, and again, when we're looking at that tumor, it's a mix of cancerous and benign cells. Cancerous cells and cells that are normal cells, and they're all lumped together into this little city of its own, really. So there's 3 stages that we think about. So initiation, when does it first begin? And then we go on to tumor promotion. So we begin to get some growth signaling happening. Growth factors are often a very big part of this, and so we'll be looking at those as it progresses, and we're always thinking it's growing in size and it's becoming invasive. And then finally, when we say tumor progression, we're really looking at it now becoming advancing toward a metastasis. And so you have this in your textbook, but take a few minutes to just see what type of cells. You don't need to memorize this or anything, but just realize that a tumor itself is going to be a mixture of these different things. And you can see right here, you've also got some blood vessels going through here, and they end up, while normally we do want that, they end up actually being hijacked and used by the cancerous tumors or even benign tumors to enlarge, because they're going to get oxygen, they're going to get nutrients, their growth factors, all these different things, and this is normally the pathway that they use to get those things. So just take a look at it and see if you can recognize some of the cells. Now, something that might surprise you is take a look right down here, because here you have immune cells that are also then going to be hijacked and used to the advantage of the tumor. It does not play by the rules with anything, and so just see it as a hostile takeover, and don't trust or believe anything that it tells you. Don't sign any papers with it. You can't trust it. Okay, so we have some of your slides may look a little bit different. I put in some animations and so forth to help you remember some of these. So in most cases, it takes more than one mutation before a cancer can develop. And so there's like about 140 known mutations. Not all of those really cause problems in the human body, but there are certain ones, and it still takes a number of those before we have something flip from being a normal cell to a benign or to a cancerous cell. And we would call those driver mutations. So they have a direct influence on whether or not a cell is going to then cross over and become a cancerous cell. There's with each type of cell type and with each type of cancer, they all have their own pathway, and so we can't just say, well, if you get 10 driver mutations, then boom, doesn't matter who you are, you're going to become a cancer. It doesn't really work quite that way. It's different for each type, and that's another reason we want to really watch these, and we want to get on them right away, because some it doesn't take much to flip it over, and other ones are more aggressive. And so once they do get started, we want to put the brakes on that right away. Now, we talked about how cancers, they play dirty. They play dirty. And as these mutations happen, we actually then begin to see them cause the surrounding environment, their neighbors, to actually give them an advantage over the rest of the neighborhood, so that they may have started out all pretty much the same, but before it's over with, they're going to be taking up all the services that are given to that neighborhood and using them to their advantage, and they really don't care what happens to everyone else. They break up these different hallmarks of cancer. So the hallmarks of cancer are going to be the main emphasis of our lecture today. You don't need to memorize which group they're falling in, but just think about that it should make sense. So there's 4 main groups that end up being the instigators and promoters of cancer. One, of course, is the genomic alteration. So changes that happen, whether it's inherited or it's an epigenetic change. It happens after you've received the genes from your parents. That will then cause one of these separately. Um, so, so we'll just keep moving here. Secondary, so these are primary causes. You're affecting the DNA of the cell. Secondary, and when you hear that someone has a disease that is a secondary, it's primary hypertension or secondary hypertension. Whatever that is, you're saying that it's not the primary source of the problem, but it adds to it. Okay, it causes it to happen because of something else. And in this case, we're going to look at increased blood vessel growth, now accentuating this primary problem. Taking the nutrients and oxygen and now using that to further this growth. Okay? So don't get too caught up in those. It's just a way of categorizing them. Other things that happen as this progresses is that we actually develop problems with being able to destroy abnormal cells. So the tumor itself develops ways to resist being broken down, much like what we talked about with bacteria, with viruses. They develop ways, they become resistant. So tumors sort of take a life of their own on. They almost become alive. They're not a microorganism. But in a sense, they operate the way that microorganisms would, in that they know someone's coming to get rid of them. They know that someone's coming to take them down. And so they begin to build defenses so that the normal mechanisms that would keep them under control don't work on them. And then again, this is why once they get out of control, it may be very difficult or impossible to stop that process. And then finally, we just look at what ends up happening once they have finally achieved their ability to invade and particularly metastasize. That this is sort of like they've been building up to this. This is the culmination. They've been building up to it with the other three categories, and now they are taking over. They are taking over the neighborhood. Alrighty. Very important, please. This is where we're taking these from. There are 10 of them. Technically, they say that 8 are hallmarks and 2 are signs or whatever. Please just consider them 10 hallmarks and be familiar with them. And really, I'm going to be outlining between Dr. Newman and myself, outlining most of what we want you to know from this cancer topic. You will find much of this in these PowerPoints and lectures. So this is a bit of an exception. Your textbook really goes in the weeds in a lot of this stuff. And so if you're going through and doing your readings and you start getting into, they get so specific with some different types of cancers and the receptors and so forth. And that's just way beyond our scope and what we can handle in our course. So pay attention to these, go in the textbook, look for clarification, but a lot of what you're going to need in the cancer topic will come from these 2 PowerPoints. Alrighty. So what I try to think about, what does it mean to me? And you have your own images of maybe how this is working. But I see cancer as kind of this bad neighbor who sort of sneaks into the neighborhood. And we're always going to remember this. The hallmark is uncontrolled cell growth or proliferation. And so what it does is it's got to find a place to live. Okay, so it finds a place to live, and when it initiates, when it first gets started, it may look like all the other houses on the street, and it's just Mr. Rogers morning. How's everybody in the neighborhood? And so when we're at this point in time, maybe nothing has happened just yet, and these all look pretty much the same. So we have cells then. These cells are called proto-oncogenes. And so proto-oncogenes, if you think about what a prototype is, it's the initial, it's sort of the sample of. And proto-oncogenes are good. They're pro-normal, okay? And what they do is these are genes that help control and make sure that as the cells divide and go through their growth, that they do it the way they are supposed to. They play by the rules, and if there is damage or if there's a DNA defect or something happens to them, then we're going to say, you know what, we have to, I'm sorry, you're just not following the rules. Let's take you in, get you checked out, see if we can fix you. But if you can't get fixed, then we're going to have to break you down and get rid of you. Okay, so we're maybe go through some apoptosis there to save the rest of the neighborhood. We can't just let you go on. So it's a safety feature. So proto-oncogenes are good. They're the HOA. I like that. That's great. All right, that's great. I like that. Okay, so what ends up happening, though, is that you end up then switching over to this oncogene. And an oncogene, if you look at the beginning of it, oncology or oncotic genes, these are now cancerous genes. And something has happened where they've mutated or they're not using the checks and balances that they were supposed to use. Yes, it's a good idea to make certain parts of the body, but when we have enough of it, we need to stop making it. We do not want them to continue to be made. And in these cases, that switch has been turned off, and they just continue to go on, and now we have this uncontrolled cell growth. And then we're also going to see the cell's not only going to grow, but it's going to begin to reproduce itself, and it's going to look like, so we're going to have a whole neighborhood surrounding this. They're going to be popping up all through the neighborhood. And if you've ever seen a neighborhood or gone and visited a neighborhood that you lived in as a kid, and maybe it was just the most wonderful neighborhood, but then maybe something happened and it started going downhill. Or maybe you went and you visited one of these old houses. That's sort of kind of what happens, and it begins to kind of spread to surrounding cells then. One of the things that happens in these mutations is, going back to cell signaling, is that these cells, many of them, begin to put out things like growth factor. They begin to send those out, but guess who they're for? They're for me. They're auto growth cells. And so they begin to growth factors. So they make it, but they don't share it with the neighborhood. It's all for them. It's like a trick-or-treat nightmare, okay? Which probably is a nightmare anyway. So anyway, they also can take advantage of paracrine signaling. So cells that are next to them that are normal or benign, they can also take advantage of what they're sending out, and they use those also to help stimulate their own growth. But autocrine is one of the big ways that they continue to grow while they let the neighborhood get worse, get smaller, and get wiped out. It's all about them. Alrighty. When oncogenes are activated, so some of the things that can happen, and I really advise you before you study this chapter, and I know probably most of you haven't done this yet because you had your exam this morning, but if you will go back and go through the genetic lecture and have chapter 2, that genetic chapter on hand, go back and take a look at what are these different mutations, because you're going to need to know those for one thing, right? But these are going to now pop up in cancerous developments. And so these are some of the things that commonly happen that help to promote what we call sustained proliferative signaling. So we talked about autocrine signaling, so sending out something like growth factors, but other things that can happen. We can actually have genetic changes that change the message that goes out that tells the cell what type of proteins or what type of function it should do. And so some of these that we commonly see are point mutations, and it works on a specific gene. That's okay. You don't need to know that specifically, but just know that point mutations are one contributor to certain types of cancers. Translocations, Burkitt's lymphoma, I think that Dr. Newman has a picture of that, but you'll see that in a lymph, so it's a lymph tumor tumor, right? And you're going to see excessive growth, but it's based on a genetic change that happens in the cell. And then something called gene amplification. And with this, you end up taking one specific, say, a receptor, for example, as we have in this picture here. So normally in the breast tissue, you would want to have a particular type of receptor. This is an HER2 receptor. And this actually, one of you mentioned about estrogens. And so this is a human estrogen receptor 2. That's a particular type of receptor. And with this receptor, you were supposed to only develop really, say, 4 receptors, okay? Or maybe this goes all around, but they're spaced out. There was a limit to how many you should be able to actually attach to the estrogen. But when this cell is being formed, what it ends up doing is it's saying, we're not going to just make 1 or 2 of these receptors. We're going to make tens of them. We're going to make hundreds of them on each of these cells. And so what ends up happening is it has a lot more receptors. They might also be much more sensitive to estrogen. So the estrogen may be able to attach to them more easily so that they begin to signal then the nucleus of the cell. You have amplified the number of receptors. You have amplified their response, their sensitivity and response to estrogen then. And that's just one example of what that would look like. So again, do we need some of these receptors? Yes, we do, because if not, this breast cell is not going to develop and mature and be able to do the function that it was created to do. But there's a limit. We need to have some boundaries, okay? And as you know, cancer has no boundaries. And then there's another class that is extremely important. So now we're going to switch over from looking at really, I should have had that section in there so I know which section we're in. But we're talking about now ways that we're able to avoid a checks and balance system again. So when tumors are present or when they try to develop, we have certain genes called tumor suppressor genes. And you could call can be also called anti-oncogenes. So we had pro-oncogenes, which could develop into normal cells or cancerous cells. Then we had our oncogenes that are oncology, they are cancer cells. And now we have these tumors, but we're having checks and balances. We're going to now try to get rid of these, or we're going to try to prevent these types of things from developing. And their job is to, as you can read here, regulate cell cycle, slow down, say there's a limit to mitosis, okay? There's a limit now. You can reproduce, but only so much. Don't go crazy, okay? So this is like the mom who has the 9 babies at one time after having fertility treatment, okay? So that causes a problem, right? And then it will stop cell division. So this is one of the big checkpoints as the cell is going through its development, its life cycle. It has those check places, we'll look at a picture in a second, that says, let's do a quality assurance. Let's do a quality assurance here, and let's see if we should let you go to the next stage. And if not, let's look at what kind of damage. We're going to just kind of pull you off of the assembly line right now, though, okay? We'll let everybody else go forward, but, you come along with me, and let's go see if you need some repair done. And this helps to prevent mutations. It also helps to prevent those mutations from being passed on to daughter cells. So what happens in cancer cells is it's like a switch, and it switches some things on, like in gene amplification. It turns that switch way up, so way more happens with it. But then in things that were supposed to keep things in check and slow them down, it turns that switch off, and it ignores them. It does not allow them to do their job. And so problem. So unregulated cell growth. Now there is a particular tumor protein called P53. P53, and you'll see this in your textbook, and it's the source of, it's a tumor suppressor gene that has widespread power, and you just turned off the power switch to it. Alrighty? Now the good thing is with tumor suppressor genes, you need to have, this is something that is, it's really more of like a recessive trait being passed on. So you're going to get one from your mom, one from your dad, one tumor suppressor gene from each parent. But in order for this to happen, in order for this tumor suppressor gene to be inactivated, both of those copies, mom and dad, must be inactivated by the cancer. So it's a two-hit mutation. If you don't have both of them inactivated, then the one that is still active will take over and become, it'll do the job. Once both of those are present, though, it's just like a recessive trait. Then if you have both of those present, then it will be triggered. All right? So most of the time, even if people inherit one of those, they just have one, so the other one is still good and can carry on. The problem is that if you have already inherited one of those mutations from your mother, your dad, the gene he passed to you, is normal, you still are looking for that second mutation. And so you're at a much higher risk for developing that cancer, because you can have then what? Epigenetic triggers that happen. And certain types of cancers have different triggers, okay, different mutations. And if you've inherited the one, you're going to get it, you're going to go into that much easier than someone else. So let's say, for example, that you have something like melanoma, okay? You inherited one from one of your parents, but you didn't inherit it from the other parent. However, we know that one of the triggers to melanoma being started, you have the gene, but excessive sunlight exposure or UV radiation will be a trigger. Well, for that person, that's then the only other trigger they need, and boom, melanoma takes off. And it's a serious and deadly skin disease. Alrighty? Okay. So these kind of work together then. So all these things that we're talking about, they work together then to make it to where our DNA and our genes become unstable, or instability. And we come right back here, and this is right in chapter 1, remember, and we talked about that as we enter in so we're not talking about germ cells, we're talking about autosomes, we're talking about the rest of your cells going into the mitosis cell life, the cell cycle. And so as they come in and they begin this right after mitosis, but they have this first checkpoint. And this is where we're going to do a quality assurance check, right? So we're going to be checking each one of these cells as they're beginning to be developed, or the genes and so forth that are coming through here, getting ready to go through mitosis. We're going to do a check to see if those are working. If it passes this checkpoint, it's going to go on toward the second checkpoint. So there's a 2-checkpoint system here. If they fail either one of them, we might be able to fix them, and this P53 is one of it's the sort of the poster child of what we call caretaker genes. So the P53 is one of the genes that we would be using at these checkpoints, and it actually is involved with repairing DNA that we've discovered here it's not up to snuff. There's something wrong. So what happens though if we turn that off and the cell then goes on to the next one, and it then maybe passes this one too? recycle the contents, the products of the DNA and so forth, but we're not going to let it reproduce. Think about if both of these, though, both of these checkpoints are bypassed because the switch was flipped off. Okay, not flipped off, that turned off. All right, that sounds bad. Okay, so if they're not working, alrighty, so then we end up then going into mitosis, and then we're going to now take damaged cells, and as each one of these then mature and go into mitosis, along the way, there's going to be more mutations, and so it goes from bad to worse, and the mutations till finally you end up with this cell that looks nothing like the original cell because it now has mutated continually, and nobody's catching it and nobody's stopping it. All right, so very, very important for this. Okay, and this is from your textbook. I know you don't have, I just threw this in. Please just take a look at it. They purposely, yes, this is a smiley face and a frowny face up here. But if you just follow this through a little bit, this tells you a little bit more about, just kind of shows you visually how this P53 would work and where it's supposed to be doing its job, and the different things that it should be helping to do to protect cells and tissues from progressing into things like cancer. But when these do not work, then this is what you're going to end up with. This large, bizarre-looking monster over here. Okay, mutant cells, mutant Ninja Turtles or something, right? Okay, they're actually pretty cute, I think. Alrighty, so other things that we can do is, and again, this comes from your genetic chapter, so it may have epigenetic silencing, so you want to go back and look at what do those terms mean, turning them off, what type of mutations might you have, and looking at the promoter regions of the genes can then be affected, and we can either stop them from going forward and developing the full proteins and so forth that they're trying to develop, or we can switch things out so that they never stop building, and then it becomes this mutant looking cell. Alrighty, so as we begin to have more of these malignant cells, we say that the chromosomes, we went from the genes being an unstable environment, now the chromosomes become unstable, and this then begins to create some of the problems that we see with some of your look at some of the children and the examples and exemplars that you saw in the genetics chapter, but now thinking about this can also lead to different cancers. Now, a big part, as we said, was that these cells, these cancerous cells, they don't know when to stop. They just continue to replicate and reproduce. Cells in the body are not intended to do that. Even cells that have a high turnover, such as epithelial cells, there is a limit to how many times those cells will replicate before they need to. They've gotten old now. They've gotten senile, cell senescence, and they really need to maybe undergo apoptosis, and they've done their duty and they've lived their life cycle. It's time to bring in some new, younger, and less likely to have had mutations over their reproductive lifetime. And you really can compare it a lot to whether humans or animals. This is why we begin to see some of the problems we see as maternal age in particular gets up to the higher limits. Usually we're saying that, beyond 40, now you have old egg cells, and there's a lot more chance that these are not going to divide and multiply the way that they should. They're going to undergo the changes with age, just like in a house, right? The wiring's not going to be what it was, the plumbing and so forth, the insulation. So every time it goes through this, there's probably going to be some mutations along the way. How do we keep that from happening? Well, one of the boundaries, one of the stop points that are on cells is that they have something called telomeres. And these are caps that are on the end of each chromosome, and they are they're put into place so that after they have, depending on the cell, after they have reproduced, they get shorter and shorter, and then finally they're not able to reproduce anymore. Okay, and they lose. Initially, they're held in place and they're allowed to be activated by an enzyme called telomerase. And this is what it's doing. It's blocking this. We don't want cells to live on forever. Okay? It's dangerous. So what happens? Well, we know cancer is not going to play by the rules. Well, guess what? The cells that become cancerous can activate telomerase again. And so it makes it where those caps, those telomeres, no longer work, and it allows those cells to continue to go on and reproduce. Okay, so you can actually, so imagine if you're in the hospital and you're an OB nurse, and you walk in, and now it would be one thing if this was, the family brought in this baby. This lady is 101 years old, if you can see on here. If someone brought in a baby that was just born so that grandma could see the baby, as she enters her last moments, right? Last part of her life, end of life. But what if she just delivered this baby? Okay, so you think about that. Is that going to be a good idea? Even if she's 50, 60, 70, but definitely at 101. It's, no, no. And the concern would be, you'd be concerned for both of them. But we know that there's going to be a lot of genetic changes, and we'd be very concerned for this child now and what they might have now inherited because of this lifelong of potential mutations. Look at this. In 90% of cancers, telomerase is restored. And this is one of the reasons it develops this immortality of reproduction. Okay? Yeah, it is spooky stuff. Alrighty, and this gives you a picture of this. It doesn't necessarily work in every cell, but especially in stem cells, germ cells, cancer cells, and we see this telomerase activity. So this should not have been happening, and now they're going to continue on. Okay, so you can look at that. Should have had a cap, and we said, great work, thank you for your service. You can stay, but you can't have any more kids. Okay, not so with our cancer cells. Now, I was saying earlier that when we're looking at cancer, we want to think back on the inflammatory process. Okay, so we need the inflammatory process as part of our immune system to attack fungi, bacterial infection, microbes trying to get in. But the same process again gets hijacked by cancer cells, and because they're now distorted, they take things that were meant for good and they twist them and they use them for evil. And it is evil. It's just a bad, just a bad thing. So what they do, think also wound healing, right? We said the inflammation process is important and necessary for wound healing. It has to have a blood supply. And then we also said that in this wound healing, we're now going to begin to see angiogenesis or angiogenesis, however you want to pronounce it, new blood vessels growing, and that was a good thing when we're trying to heal a wound because it's going to bring oxygen, nutrients, growth factors, all these different things, and it's going to help to get rid of some of the dead tissue and waste that we're trying to get out of that wound bed. Well, think about now, if I'm saying I've got this malignant tumor over here, okay, and I can actually send out and make use of, here's my main blood vessel, Here is a blood vessel that's traveling nearby. Okay, not unusual. We saw in that picture that in the stroma, there will be blood vessels that go through there. But what if I took the growth factors that were supposed to feed the normal tissue and I make it grow more around my cancerous cells? And then I do something else. There's a particular growth factor, and this is called vascular endothelial growth factor. Okay, so think about it. Blood vessels, vascular. Okay, endothelium. Okay, these are the cells that line this. So one of the things that it does, though, is just like in the inflammatory process, this triggers these endothelial cells to become more vascular. It increases vascular permeability, right? So now this tumor receives more that's flowing through here, but it does something else. It allows then tumor cells to seed and pass through into the blood vessel and then begin to what? To metastasize, and now it's going where? To my bone, liver, brain, wherever that particular type of a cancer cell finds friendly receptors. So it looks sort of like for open houses, it looks to see if this is going to be a friendly neighborhood for it, and then it takes it over. Okay? So again, thinking inflammation, thinking how it relates to wound healing, but twisted and evil. Okay, this is just sort of extra. There was something called the Warburg effect. So looking at now, so we talked about blood supply, but think, what would that blood bring? Well, it's going to bring things like oxygen and nutrients. Okay, so normally blood supply is going to do that. That's one of the big reasons we have to have it. So in this Warburg effect, and these are kind of, both of these are sort of hypotheses. How do cancers proliferate? Do they actually work exactly the same as normal cells do? Do they have other hidden agendas and pathways that they can use? And so one, the Warburg effect is saying that it prefers, cancer cells would prefer to undergo normal glycolysis. So we think about that picture, right, of a molecule of glucose. In aerobic, oxygen is present, then it enters into the mitochondria, and we produce a lot of ATP, okay? Would that ATP help cancer cells then to grow even more? Absolutely, it would. Would it want those extra nutrients, oxygen? Yes, absolutely. So it kind of goes hand in hand there, the oxygen with the ATP. There is another theory, though, now, and this is just a very general thought process, is that it could go either pathway. It no longer is super picky about how much oxygen it needs, or whether it has an ideal environment for nutrients. So we've talked before about how there are some bacteria that really prefer an oxygen-rich environment, or they really prefer an anaerobic environment, no oxygen, low oxygen. And we think of Clostridium with that type of an environment, an anaerobic environment. So but then there are also bacteria that they don't really care what you do. You can go plant them out in the yard, like here in Texas. Some plants need a lot of water, but we have I always say this wrong, the xeriscape plants, right? And they don't require a lot of water. And then you have like succulents, cactus, that if you give them a lot of water, you're going to kill them. I know, because I've done that. I've killed all of them. But so there's some thought, though, that at least with some cancers, that they can adapt, and you can starve them of oxygen, you can starve them of food, and they're still going to continue on. So just some food for thought. Okay. All right. So looking at reprogramming some, again, metabolism. So what do? Well, they actually then, whatever is available, what are they going to do? These oncogenes, these cancerous cells, they can what? They can be microenvironment that they live in, and they can steal food and oxygen from those other cells and use it for themselves. So they're very manipulative. You can't trust them. Again, don't sign on the dotted line because they're not going to come through. It's all about them. Switching gears a little bit now. So we said that when we were in the cell life cycle, that we had those checkpoints. And one of the results is that when something defective is found in you, then we don't want you to reproduce anymore. And in fact, if we can't fix the DNA, we'll try to repair it. But if we can't fix it, then we're gonna turn on the self-destruction. All right. So I don't know if you watch Star Trek or something like that, but they have those self-destruct things. Okay, we can't survive this battle, and so we're going to program the computer to self-destruct in 60 seconds, and everybody needs to jump off. And so what it does, though, is it says the controls, the P53 tumor suppressor cells, they're saying, okay, this is not good. We always want to put out a great product out there. We want everybody to say, we make the best cells. So we're not going to put out defective products. So I'm sorry, but you're going to have to self-destruct, and we'll take some of your pieces and we'll make a new cell. Okay, it's not totally over for you. We'll use your parts. Cancer cell says, I don't think so. I don't think so. I do not wish to die, and I'm not going to self-destruct, and this is one of the ways that I'm going to resist you. And so you can see in this picture that instead of undergoing apoptosis, it keeps dividing, but it's defective, and it should not divide. Right? So we talked a little bit about inflammation, but I want you to make the tie also that when we have people who they've gone through acute inflammation, they've had an injury, they've had a wound, and in that acute inflammation period, about a 2-week period is considered acute. As they progress beyond that, and especially as it goes on now for months or years, keep in your thoughts that this chronic inflammation actually begins to trigger the development of cancer in some cells. Okay? It makes some of these tissues more receptive to the changes of going from a proto-oncogen to an oncogen. And so, do we deal with a lot of inflammation with our patients? The more that you study these and the more you look at these exemplar diseases, as you're taking care of your patients, go back and see what is usually the cause. And wherever you see that you've got a chronic inflammatory process going on, ask yourself, I know that creates a lot of problems to the body, to joints, to linings of the GI tract and so forth. Could this also lead toward a cancerous situation? What do we need to do to try to get this chronic inflammation, if not stopped, at least under control? Okay? One of the things that we see happen is something called H. pylori, and this is peptic ulcer disease. We used to think that we didn't know that H. pylori was the biggest contributor. An infection with this was the biggest contributor to peptic ulcer disease. And so when we found that out, so we'd just been treating the high acid, trying to repair the damage, keep people from eating, spicy foods and things that would trigger acid production in the stomach. But when they found out that this is actually more of a bacterial underlying cause, then what could we do? Well, how about treat the cause? Okay, so yes, we're going to treat the symptoms, but wouldn't it be better to actually get rid of the cause? And so now the whole goal of treatment and the whole pathway that we used focused on killing out this bacteria and then also treating any damage that might have been coming along with that. All right? So medicine changes, the way we treat patients change with the science that we learn. Okay, so still talking focusing on tumor promoting inflammation. We'll get to a little breaking point here, and I'll let you guys take a quick break. Because I don't want to keep you here all day. I know you're tired. So there's something called a tumor-associated macrophage. So we're thinking cells of the inflammatory system. Well, now we have macrophages, though, some of which are associated with tumors, and that they actually help to promote survival of tumors. Okay, we're not going to go in great depth with this, but just know that this cell is involved. And when we see this, there's different types of cells. There's M1, M2. There might be others, but we definitely have the M1 and the M2. So we see the M1 more in acute phase. We see the M2 in wound healing. Well, when we have tumors and we do a biopsy in them, and we see that there are a lot of M2 types of macrophages presence, this tells us that we're probably going to have a worse outcome or prognosis. All right? So we do need those M2s when we're doing wound healing. But when we're talking about tumors, did we need this inflammatory process going on? No, we didn't need it. We're not trying to heal anything, and it's hijacked it and it's using it to its advantage. It also then leads to less of a cytotoxic cell. So T cells and natural killer cells are no longer able to really act on these tumor cells. It blocks them. And it actually, again, the cytokines that they're sending off to bring help to the area, like they would be in an infection or in wound healing, it actually uses those again for itself to get even bigger and go even further. Right? Alrighty. So we talked about inflammation. Let's look at immune system, because it just hits on so many different parts. So another section of the immune system, though. So even the normal immune system should normally help to protect against cancer, right? The ways that it's going to do this is going to be watching for them. We have our cytotoxic T cells, natural killer cells. That's one of the types of cells that they kill is cancer-infected cells or malignant cells, virus-infected cells, and cancer cells. However, if we think back to the slide before that, we can see that it's not always going to be effective, and that they may have the ability to actually turn off those protective surveillance cells. Alrighty, so just kind of looking at this, and I think about how viruses kind of sneak their way in, right? And if you think about them attaching to the T cells, and think about especially with HIV, I think of. So it's going to, at the very end, it's going to attach to a receptor cell on the cell surface. It's going to go into the cell, and then what does it do? It reveals who it is. So it's an imposter, and it's saying, no, I really need to come into your house. I need to come into your house, and don't worry, I'm harmless. And so I like this little cartoon here. Can I take your coat? And this is a conference for thieves and con artists over here. So it works kind of the same way. It comes in, and it's really a con artist. So you can let me in. Don't worry about it. But then once it actually gets in there, it begins to do some of the things that it said it wouldn't do. It lies, and then it takes over the cell and the surrounding area and uses it for itself. And when it's in there, it's a lot harder for it to be seen, right? Some of the ways that it keeps itself from being seen, it has the ability, if you look in this top one, this top section here, you have it in your textbook, this is how it's supposed to work, all right? So a tumor cell has some tumor antigen in it, and it's supposed to present it on an MHC molecule. And then it presents a piece of it out here that then the T cells should be able to align up with it, should be able to recognize or bind with it, and then destroy it. So some pathways that it might use to avoid this from happening, from avoid letting the T cells do their job, is there's no antigen to present. So certain types of cancers, they're going to act differently. If there's nothing to present, then there's nothing to flag the T cell that you got a problem here and you should attach to this. It's not going to recognize it. In this section, maybe something else is missing. A certain type of MHC, the presenting portion, it's not there. It's inactivated it. Well, if I can't present a piece of this on the cell surface, again, it doesn't trigger the T cell to respond because it can't see inside the cell. It doesn't work that way. So my cytotoxic T cell doesn't see that there's anything wrong with this cell, so it leaves it alone. And then there's a third way, in that it goes through all the steps that it was supposed to do. So we have our tumor antigen here. We presented it on an MHC molecule, and then here is our piece of tumor molecule, a piece of it out here that should be recognized, but guess what it does? It cancels it out. It gives it a negative feedback saying, no, nothing to see here. It inhibits its blocking. It tells it, no, just turn yourself off, nothing to look at here, okay? So again, very sneaky, deceptive, but a wide variety of ways to avoid this type of destruction as well. All right, almost to that breaking point. We'll take a little break. Yeah, it truly is. I see it as having like this personality. It's just, it truly is an evil being. If it could be a being, it would be an evil one. All right, so finally then, what do we say? Invasion, metastasis. We go through all of these things. We let it grow. We let it multiply. We steal everything from around us for me. And then we finally get to the point of invading tissue around us and jumping on into the bloodstream or into the lymph system and spreading now. And this is where we've got our real problems, right? Okay, so when we have localized, we say that it's localized, right? And this is a picture of a breast tumor. And so still in the local area here, doesn't look like it has passed through to this basement membrane, but it's about to. And we know that what we've got, ducts here, milk ducts, and so forth. We've got blood vessels, and so at any point in time, this could cross the membrane, the basement membrane. It could go into the blood or lymph, because we've got a great lymph drainage from the breast tissue, which would be good, because if you have someone who's nursing, could easily get an infection. It's going to, and we want to be able to drain that off into the lymph system. Well, unfortunately, cancer will also follow those routes. So then we can see where we've got some local metastasis, and really watch for this in Dr. Newman's lecture. So we have some local sites it's going to go to, lymph nodes, right? Because it's going to go right into that section there, and then can be passed along, but it's not being destroyed. It's spreading the cancer. And then we finally end up going, getting into the blood supply, and ending up traveling to a distant site. So think right here, bone, way down here, liver. You're not talking about the breast tissue itself. You're not even talking about the surrounding lymph structures. Now you've gone to a remote area. A very bad sign. And as we talked about earlier, if I were to get a sample here of a bone or liver mass, but I see, you know what, this looks like breast tissue. This is like messed up breast tissue. Well, guess what? You probably have an original lesion, a primary site, and this is not new liver. It's a secondary lesion. Okay, very scary. Alrighty, and so there's also a little bit of a possibility that as they're going through this stage, so we said that stem cells, as they're progressing forward toward a more differentiated state, they don't turn around and go backwards. But we said that cancer cells, they actually have the potential to do that. So they can stall themselves and change up the DNA a bit. They also have the potential to return to a more stem-like state. And then they have that potential then, as a cancerous cell, to go on and to invade different types of organs rather. So some of the things that we're going to see then when cancer is what kind of things would we look for? What's going to tell us that someone might have a cancer? And so these are some things that your patient, signs and symptoms your patient might come in with. Again, fatigue being a symptom, something like anemia being a sign that I can actually measure and see. So one of the things, though, is something called paraneoplastic syndromes, and these are actually a group of symptoms. It's not directly caused by the cancer itself, or the mass of the cancer, but it's triggered by cancerous things. And I'll see if I can come up with a good example for you of that. So I'm not going to be testing you on paraneoplastic syndromes, but you might hear that mentioned. But some of these, and if you're looking back at what do cancers do, these are some of the things that you'll see happening. And as we see them taking over the bone cells, if they're triggering anemia, they're affecting blood cells, or they're affecting the bone marrow where we're going to have blood cells made, then anemia, as I don't get enough oxygen or red blood cells, hemoglobin, fatigue, in fact, the most common complaint. But it's so vague that someone coming in and just saying, oh, I'm really fatigued, I'm really tired lately. Well, that could be anything. That could be COVID, right? That could be that you don't sleep enough, you don't eat it. It could be being a nursing student, right? So we have to look at these various things and take the pieces together, and it's not necessarily an easy conclusion or diagnosis to come to. And then when you think about how many different types of cancer there are, so we would want to be, they could have various manifestations. So there are certain ones that really flag us as, hey, we think we're probably looking at a cancer. And one of the big areas would be looking at changes in white blood cells, lymph cells, red blood cells. Watch for anything bleeding that is unexpected should always be a red flag to you. And they don't make as big a point of that in the textbook as I think in practice we do. So if you have somebody who appears to be menstruating, they have blood from the vagina, but what if it's the 100-year-old grandmother? Okay? What if it's happening between? Well, it doesn't mean that they have cancer, but you need to look at it. Somebody having rectal bleeding, any bleeding that we don't have a good explanation for needs to be investigated further, because that's one of the signs that we see with cancer. And it's as it's invading through and damaging, it can damage blood vessels also. So, a lot of different reasons. If it was in the GI tract, it could be breaking down that the walls of the GI tract, and then blood escaping from blood vessels. So always be watching for that. Alrighty. Dr. Newman does more in this, and I think it's pretty straightforward. I'm sure your PALS leaders will be addressing this also. So there's a couple of different ways that we do staging, and it really, if you just look at it, a stage 1, being no metastasis, looking also at like the lymph areas. So like in that breast tissue picture that we looked at, and then finally stage 4, distant metastasis. And so by this point in time, we don't really know how much time you have, and a lot is going to depend on what is the cancer. So things like pancreatic cancer are often discovered very late, maybe not discovered until stage 4. Until stage 4. And then by that time, I was reading about an actress. Actually, my husband and I were watching a movie the other day, and I was just Googling who this actress was and where she was now. And it was saying that at a pretty young age, young for dying, I know it's not young to you guys, but she was like in her late 50s. She died, and so I was looking to see what happened. Turned out she was diagnosed with late stage pancreatic cancer, as is often the case. Pancreatic cancer is generally very aggressive, and by 2 months later, she had died, and 2 months earlier, she didn't even know she was sick. Okay? So we look at things like acute lymphocytic leukemia. That's another one that we see, just very, very rapid, very fast. And again, it's the kind of thing that when the lab results come back, and on some of these they can tell just by taking doing a CBC. And the numbers are so out of control, out of crazy white blood cell count, just totally out of control, that these patients, we will track them down. We will find them and get them to the hospital. And so, because they have to begin treatment immediately. And we have no idea if it's going to work. So there's not like, we'll come back in a week and you think about your options. It's like, you start today, or, we don't know how much time you have, but not much. The other is the TNM, so T for tumor, nodes, and then metastasis. So two systems that you might see used together, commonly used together, that gives us an idea. And this is basically this is one that combines the two, so you have the stages. They're really actually looking more at the TNM, sorry about that. And so you can see how they would grade that. And again, so here's our tumor, right? And here now you're breaking out, you're going through the basement membrane, and you're starting now ready to travel other places. Nodes, talking about lymph nodes, okay? So nothing found in the lymph nodes, a small amount, many and diverse lymph nodes, much worse sign, okay? Then metastasis, as we've already discussed. So be familiar with those. If I test on these, it'll be a broad, it'll be pretty clear. So you don't need to necessarily sit and try to memorize each of these, but just be thinking, if 0 is low and a good sign, and 3 is a bad sign, then know what they represent, and are you in a good position or a bad position? All right. And this also brings up a point, because one thing that we will do, especially with things like breast lesions, is that they will go ahead, if they find out that yes, it is a malignancy, then they're going to go to the lymph nodes and remove at least some of the lymph nodes that breast lymph system would drain into. And this is why, because we need to know, is it spreading and how far is it spreading, so that they know how to plan treatment. As you're taking care of patients who have had a mastectomy, had lymph node removal, you want to always remember that we will not use that arm anymore for any procedures, because a lot of, or all, they probably can't get all the lymph nodes, but many of the lymph nodes were taken out. Well, the hand is going to drain, the hand and the arm are going to drain into that lymph system, but now they can't. So there's going to tend to be a buildup of lymph fluid in that affected arm. Also, if I got an infection, even from a glucose finger stick, an Accu-Chek, behind B cells, T cells, and so forth, and it's not going to go there. So some people will have so much lymphedema building up, and in order to prevent that swelling, because we know swelling itself causes problems like decreased circulation, so what they might do is they might wear a compressed sleeve, like we do with putting compression stockings on people's legs who are in bed for a long time, helping to prevent swelling from building up. They might wear that on their sleeve, but absolutely do nothing on that arm. Don't even take blood pressures on that arm. Switch to the other side. We don't want to take any chance on that. Okay. Oh, goodness. Anna, that's a terrible story. And, sadly, we've gone into nursing. We're going to be dealing with a lot of these kinds of things. So, tumor markers. How do we find out if people have these? Well, there's different types of things that we call markers that we might be able to find out. So these are things that are in the body that the cancer cells actually produce. They might be within the cells. They might be circulating in the blood or in the lymph. Normally we don't, except for doing biopsies on lymph nodes. We don't really access the lymph system, to my knowledge, but we would take a blood sample. It's easy to get a hold of. Certain types of cancers, we might get CSF or cerebrospinal fluid. That wouldn't be really a common thing to do, but depending on what the problem was, maybe if it were like a brain tumor, or we were concerned about infection in the spinal cord or cancerous cells in the spinal fluid, urine. So just think about things that we normally get a hold of, but we can also check now for markers for tumors. And these just give you an example of that. So someone had asked a really good question. What do you do to find out if maybe are you that person who might have inherited something that's a serious or life-threatening cancer? Is it something that's genetically, there's a high risk of passing it on? And I put here a couple of examples of people that you can. One is an actress, another one is a news reporter. They have shared their information so that others won't be afraid to go and ask, and also to get the treatment done. So they both found out they had inherited the genetic risk. And again, they're going to look at the family tree, plot that out, and then look to see in genetic testing, have you inherited now these, or do you have a high risk of this? And so on both of these ladies, what they opted to do was to go ahead and get a prophylactic, a preventive bilateral mastectomy, which is a pretty big step. It's not something you want to just, I think I might be at risk, so I'm going to go and have parts of my body removed just in case. Pretty serious risk to know it's out there. And so you want to be seeing specialists, geneticists, and so forth, but they made that decision because the chances of them getting an aggressive, early in life-threatening cancer was very, very high. And by getting rid of those, and my guess would be if they are estrogen dependent, which with it being breast cancers would be a real possibility. And so you're going to also maybe see patients who either have those cancers or have a high potential for having them, have things like hysterectomies with their ovaries also removed, and oophorectomy. Anything that might contribute growth factor to a specific type of cancer. So you can see you're going to be dealing with a lot of not just the physical aspects, but imagine if someone is telling you, yes, you have this. And imagine if it is maybe you don't have children yet, and they're saying you're going to have a hysterectomy. You're going to have your ovaries removed. So there are a lot of emotional things we deal with, and so all the things you're learning in your other courses about professionalism and how we communicate with people. Sometimes it is as important as being skilled technically at a variety of different ways, genetic testing. I'm not going to test you on treatments, but just so that you know that these are some of the ways we test for it, and then some of the ways that they're actually looking at treatment is by targeting, just like with bacteria, viruses, and so forth. Some of the ways that they're looking at is to develop by knowing what things or what portions of this cancer, what they're really receptive to, is to develop a specific immune genetic therapy. And so that's pretty exciting, actually, if you could do that instead of just having to have things removed, right? Alrighty. This is an example of a needle biopsy of the breast. We used to do these a lot in the surgeon's office I worked for. It's not a lot of fun to watch, and it can be painful for the patient. But this is what they would do: is they would take a large bore needle and go into using an ultrasound to guide. So it's an ultrasound guided needle biopsy. So usually going to do this in you have to be able to have like fluoroscopy or be able to watch the ultrasound as you're doing this. And so they're going to take a piece of this tissue, just a portion of it, a biopsy, and then send that to the pathologist. And from that point on, they would know, do we go further? Do we just watch this? What are we looking at here? Now, one of the concerns, though, whenever we do cut into a tumor, even by doing a needle biopsy, and I'm sorry, I can't always see my arrow. I'm trying to make anybody dizzy or nauseous there, is that this is a large bore needle. So like drawing blood, big needle so that those cells don't break up as they're coming in, right? And this is a solid tumor. We take the risk, if this is a malignancy, you take the risk that some of those cells, as you're pulling the needle out, you're pulling this needle back, that some cells, which could be cancerous, will then be left in the surrounding tissue. So we could be doing seeding of these cancerous cells, and that is a risk that we might have to discuss and be sure that our patient understands that. Now, do we have to know what's there? Yeah, we do. So it's sort of do the benefits outweigh the risks? Okay, but that's called tumor seeding, and that is one way that tumor seeding can happen. We talked about the markers. We can also do different types of imaging tests like CAT scans, MRIs, PET scans. And so you'll learn more about those as you're going along. And then thinking about, remember when we're saying, we also do mammograms, of course, would be very important with breast tissue. Probably if somebody is at considerable risk for that, they might have those done annually. One of the things they're looking for is calcium deposits. So going back to our first lecture, when we see calcium deposits, it's never a good sign. So calcium in the right amount circulating in your blood, good. Calcium other places or showing up like a shining bright image, bad. Right? Okay, lifestyle. Things, so we're thinking epigenetic, especially in this section. So things that we do or that we're exposed to, whether we intended to be or not, that can give us that second trigger or fourth trigger or whatever, that lead a cell to go from being a proto-oncogene to an oncogene. Different types of pollution, radiation. So we know that. When you are taking care of your patients and you take them to get an x-ray or they bring it to the room in the ICU, most of the time they're going to come to the room and they say, x-ray, get back. All right? If you're not wearing one of those badges that detects the amount of radiation you're receiving, you need to be far away. I believe it's at least 6 feet. So don't take it lightly. Get out of the way, get back, let them take the pictures before you come back in. When you're in the rooms, you're going to be given probably a leaded apron to wear, and a leaded wrap that goes around to protect your thyroid gland. So protecting the reproductive organs, protecting the thyroid gland. Be sure that you don't omit those because you're going to be exposed to these, and you're not. If we're not keeping track of it, you don't want this to be the reason that you end up developing a certain type of cancer or it affects your reproduction or thyroid. So do be careful. Other things, lifestyle, okay? Smoking, alcohol, diet, exercise, obesity, sexual practices, especially risky practices, and different types of infections. We'll take a peek at those. All right, so three major areas that what? Epigenetic, now making that second trigger, and then we see that we begin to have some changes here, noticing tumor suppressor genes not being expressed correctly, oncogenes being triggered. And then doesn't mean you're going to get cancer, but you're raising your risk every time. You're raising your risk. Alrighty, this I think is in your textbook. I'm always convicted when we do this, and then when we do the GI lecture, I feel very bad. Inflammation, all of those things. I start thinking, someday I'm gonna have to start taking better care of myself. So you start now, and I'll try to catch up at the point that I can. Please know this. Remember this picture. Everybody, when we were growing up, Dr. Cleveland and I, we joked about this, that everybody had an ashtray. You had ashtrays all over your house, whether you smoked or not, because it was impolite if you had a visitor. So the polite thing to do would be to ask, does anyone mind if I smoke? And if you were the host, yeah, that's right, yeah. If you're the host, the polite thing to say is, of course not, let me get you an ashtray. And so you'll find, actually, if you go like to an antiques place or something, if you enjoy looking at old things, that they'll like have all sorts of ornate ashtrays, any kind you could possibly think. Clay, they'd make them, like Craig is saying, in school. That's what, hey, we made you an ashtray. And so now things have changed quite a lot. But I want you to remember this picture. The number one preventable cancer, smoking cigarettes. Coming up right next to it, and I think we'll surpass it, Dr. Newman's going to address this, vaping. So if you're not convicted enough right now, I want you to think about that. If you're a vapor, if that's a word, stop now. The reason they don't have a lot of data on it is because it's recent. I'm telling you, there is no way that this is not going to end up leading to cancer, and he is so big on this. Listen to what he has to say about it. I don't have data. Actually, he does. Dr. Newman addresses it. Dr. Newman addresses it, and he's going to say, don't do it. Don't do it. All right, big carcinogen also on the marijuana. So I'll leave that to his lecture there. But there is no benefit to this. And I worked for a dermatologist for a while too, and even just smoking, pursing your lips to do the inhalation, to inhale the smoke, well, guess what? That ends up leaving lines around your mouth that by the time you're 40 years old, you won't have to tell people you smoke. They won't have to smell it on your clothes or anything, which some heavy smokers, that's not unusual. They'll see it by the wrinkles that are forming around your mouth. Okay? So if that doesn't help encourage you to stop smoking, vaping, whatever it is, please, please, please think about that. Obesity, a big factor, and what? We're talking about insulin, we're talking about growth factors. Insulin growth factor is one of the big triggers with that. And so three things, insulin growth factor, we talked about different sex hormones, particularly things like estrogen, but also then we add on adipokines and adipocyte-derived cytokines. So these things trigger it. Lack of exercise. Man, it's a good thing it's not Friday, right? I just ruined your whole weekend there. Alcohol consumption. So think think balance. Be careful. We're not going to encourage, even though maybe there's some benefit to a glass of red wine, cardiovascular wise, that's what data is showing. We don't want to encourage patients to start using alcohol, though, if they're not using alcohol. And so many issues that come up with alcohol. But I digress. Another day. UV radiation and tanning booths, just take that and concentrate it. Melanoma, basal cell, squamous cell being even worse than basal cell. And these actually can go into deeper areas of the tissue and can cause some serious damage, even going to the point where people have to have large amounts of not just skin, but muscle and maybe even bone removed because of the damage from that. So please use your sunscreen and get those things checked. You're going to be in HEB, and someone's going to be in front of you now, and you're going to be going, I don't know this person, but I've got to tell them they need to go and get that checked. And your nurses now, right, are on your way, well on your way. Let's touch real quickly on different infections that can lead to cancer. And these are ones we talked about in our infection one lecture, but especially looking at viruses. So different viruses have the potential of causing serious and life-threatening cancers. We talked about H. pylori, not only causing inflammation and so forth, but leading to stomach cancer. 75% of all stomach cancers could be on an antibiotic. It's really a cocktail of antibiotics, similar to what we do with HIV. Not the same drugs, but needing to take multiple ones to get the effect that you're looking for. Hepatitis B and C can progress onto that. And then Epstein-Barr virus. So we see HPV, thinking we're going into this a little bit more. Watch for your patients, especially who are immunocompromised, and especially HIV patients, that they are at higher risk for some of these other viruses, and including different types of, like, Kaposi sarcoma. HPV, sexually transmitted disease. Okay, see that really with viral warts, able to cause cancer of, most commonly seen, of the sexual organs. Okay, also oropharynx. This is the organism that we are doing Pap testing for, and so the Pap test needs to be done regularly. And Dr. Newman's going to talk more about those. Cancer, thank goodness, it's rare in children. We're almost done. We're going to just touch lightly on children. As I said, for the entire course, we're just going to touch lightly on this. So thank goodness we don't see it very often, but when we do see it, we see it more in the early stages. Okay, so usually looking at intrauterine. Okay, this is the most common times that we see it is embryo tumors. All right, so we might not know that they have it at that point in time, but it might be some testing for people who are at high risk, maybe by a family history. Look at this little girl, don't you just love her? So most of the time, they haven't lived long enough to have the epigenetic, the buildup, accumulation of triggers and hits on their cells and DNA and so forth. So that tends to be a little more why we see that these are actually things that are genetic or in utero. Let's see. Not too much. They could have other causes, but in general, just think about, you're not dealing with a lot of the things that we deal with adults when you're taking care of pediatric patients, because they just haven't lived that long. They haven't been as exposed to things. These are some of the types of cancers that you might see more commonly in children. They're really diagnosed in children. We see some familiar terms: tumor suppressor genes being affected by it, mutated, not working. And so you have a faulty gene, right, that keeps being reproduced. some of the changes that we see. I'm not going to test you on these. You don't need to memorize these. These are just given for your examples. Listen for them when you get to your PD course. Some things that could happen that are epigenetic or environmental, exposure to something called DES. So sometimes there are things given to help prevent miscarriage or to help control people who have a lot of symptoms like nausea, vomiting, that type of thing, not knowing that they had later effects on the children. And I'm trying to think, if anybody can think of it right now, I'm trying to think of it was a terrible, terrible deformities that occurred. I can't think of it, where people had poor limb deformity, like they were born without any arms or legs. Thalidomide, thanks, Craig. So thalidomide babies is what they fell under this category. That's what they were called, and it had to do with them receiving a medication that was for nausea, and it had no idea that it would have these profound birth defects. So other ones, I'm thinking with the DES, that it ended up affecting the reproduction organs in females, and you guys can tell me if I have that right. I think it was the DES, though. And so the good news is that the babies, survived and were born, but then they later had problems with not being able to have babies of their own. So we have to be so careful, and this is why I generally don't take care of pregnant moms if I can possibly help it, because we have to watch every drug that they're being given, everything that they're being exposed to. Don't take anything for granted. But always have to think about the developing child, and you're definitely going to be checking your drugs and looking for black box warnings. Can this be given to someone who's pregnant? So be very, very careful about that. All right, so some of the different things, same types of things that we have. Again, I'm not gonna test you on these, but you see some familiar names here. So things that they can maybe also then inherit or be exposed to. So they can also catch AIDS, Epstein-Barr virus, and so forth. All right. So good news and bad news. Good news is, and this is so different than when I was growing up, more than 80% of childhood cancers are now curable. That is huge, huge progress. Huge progress. That doesn't mean everybody is, but what a turnaround. The bad news is that they tend to be at higher risk for developing cancers. So if they survived or cured of cancer as a child, then they need to be monitored much more closely in later life, depending on the type of cancer that they had, because they are going to be at a higher risk for developing certain types of cancer in later life. Alrighty. And that's all that I have for you. I think that's plenty. But I hope that helps to explain a lot of these. Again, certainly use your textbook, look at the pictures and so forth, but between these two PowerPoints, Dr. Newman's and this one today, you really have the highlights of what we're looking for on cancer. There's just way too much information for us to cover more. And I'm gonna speak to the editors of your book and say, okay, guys, you're getting a little crazy here. Somebody really likes studying cancer, but I think you went off the cliff a little bit on this one.